So if you click this, okay, then it will take you to the next page. On this page, all those monster things, they are summarized, okay. So on this page you can see less things and a bit more organized. Here you can see the name. This is the exact name of your gene that you have to use. Gene transcript number. Every gene has specific numbers. These numbers are not going to change, okay. This is the gene code, gene number. This is a transcript, HD. Yeah, as I told you, HD38 is a human human sequence, the reference sequence that we are using. This assembly HG18, human genome 38. So this is the chromosome 9, position number this. So this size of your gene is 52,383, exact base pair position. Total 13 exons, and your gene is on the negative strand. You know the DNA has two positive and negative strands. So whenever we sequence, so we mention either is it a negative strand or a positive strand. It's nothing, it's just the complementary. Positive and negative are only complementary sequence, but they are, like, of course the gene is same. You don't need to go into that detail, I guess. Then comes the coding region. This is the gene transcription, okay, the whole gene. Coding region means only exons. When if you are only seeing the exon area, then this is the coding region. So coding region is 50 is also like 51.73, and 13 exons are the coding regions. Here if I'll open someone, I'll show you the actually gene sequence. I think all these are different things that you can see and check if you want. Then from here I can go to the messenger RNA. If I want to see it's only its protein, I can open and see its protein sequence. If I want to see gene sorter, I can open it and see. If I only want to see exact gene sequence, then first go to the gene sequence. I'll show you. Gene sequence is like this. When you open the gene sequence, this bar will come. Don't change anything. Let's see. Okay, one thing, if I want to see that exon in the upper cases mean all capital exons, everything else in the lower cases. So let's see, let's submit it. And this is the whole, how much thousand it was, like 51,000 sequence was there. So this is like the whole 51,000 sequence, okay. Yeah, that's it. All the capital, we mentioned that capitals as exon, keep them capital. So this is exon. Wherever you see the capital, they are exons. So this may be the last exon, number 13. If I'm going to the top, like this one, first exon. Okay, and always the start codon is in the first exon. ATG is the start codon. So ATC, ATG, and here let's see where the start codon starts. Sometimes it is in the first exon, sometimes it is in the second exon. Here you can see the second exon, okay. So in this data there will be 13 exons. Okay, you can check all of them one by one. If I go back again and if I change this, instead of one per gene, if I change this per region and again submit it, then it has mentioned like this is the exon 1, then exon 2, this is exon 3. So per region means exon wise they have mentioned from which, in all this region you can see there are like 13 exons. You can go back. So this is about the sequence. If you want to take the sequence for making the primers, we get the sequence from here. Okay, so from primer, so whatever exon your mutation is, so you have to go to the, suppose if mutation is like the exon number 3, you can go to exon number 3 and copy the data from, where is the capital letters, like here are the, cannot see the capital letters. Suppose if exon 2, the capital letters are here, so you have to take the data from here and copy paste the data for making the exons. But we always copy paste more than the actual region to not skip anything. Okay, let's see other things on this page. This was the protein sequence data that we opened, protein sequence data, which is 501 amino acids. So you can see this is amino acid like phenylalanine, alanine, thymine. Okay, so this is protein sequence. This is used for if you're making the cDNA and so on. This is not used in your thesis, but I'm just showing you, like, you should know what are the gene, its components, at least your gene. You have to play all these things with your gene. Okay, from here you can go directly to the gene card. Also, I want to show you some expression. Where was the expression data? Also here is showing the gene interaction, that this gene interacting with what things. Let's see this gene card. This is the gene card. You can go from Google to the gene card as well, write your name of the gene, and then all information of the gene will come. Okay, so as I told you, all these databases are connected, so you can go from one or the other and so on. So this is the name of the gene, is family. It's a uniprot site. Swiss prot is for the protein. If you want to go and look for protein amino acid structure sequence interaction, you can go to these databases. For like generics, you have to go to the OMIM. Ensemble is also for the protein. NCBI is for the gene. Okay, this is the summary. From here you can get the summary, what is important, why your gene is important, what pathways it is involved. Also the references, recent references are given here, so you can mention it in your thesis summary. Okay, so you can read them from the protein point of view. What is the protein like, its structure, what interactions it involved. This picture you can copy paste in the thesis if you think this is more clear than the UCSC. And this is the structure 3D PDB. It's a protein database 3D picture of your gene. This is, you can see, model, the different models which are present. So all this different type of information you can get from the gene card, and so on, so on. Subcellular location they have mentioned. Also here the pathways are given, as I have opened here as well. This pathway you can also give, that this gene actually interact with which genes, directly, indirectly. Okay, gene interaction is also important because actually in nature anything alone is not working. So which gene is interacting with more genes? Suppose if this is your causative gene, and you can see it is controlling so many other genes, and if you just Google those gene names, you will get to know what they are doing. If this gene is involving with neurological disorder, and these are also involved in one of these disorders or some important pathways, so you can mention it. You can give this picture in the literature review, add with the reference, and you can mention in the discussion that this gene has so much important in interaction with other pathways, other genes, and this is how it is working. Okay, all this information you can get from here. So it's up to you how much you elaborate your thesis, how much you are playing, how you are playing with your data. One more thing, you can play with your data by taking AI help, that how this can be used in your thesis, but you are not supposed to copy paste, because in your thesis plagiarism AI plagiarism must also be below 90%. It means if it is 90%, 19%, your thesis will not be accepted. This is another diagram showing different gene interactions. So this is how you are elaborating your result. This picture is showing the gene expression. So here you can see that nervous is green color. That is it is expressing, this gene is expressing in the brain, cortex, cerebellum, and the spinal cord and the tibia nerve as well. So it has a high expression in all the brain tissues. Plus it also has major expression in the all the vital organs like colon, kidney, liver, lungs, spleen, stomach, all the vital organs. Now here down the disorder of the ALDH, it has mentioned what disorder is already reported. Okay, so down there you can find like alcohol dependence, anaerobic meningitis, lung cancer, colorectal cancer. These are the reported disease link of your interest gene. So whatever your gene was, you can come here and see which disorders they are linked. Here you can see all the publications and data. Okay, so you can start your thesis by playing with these tools and getting data, retrieving all the data from these sites. I hope I have enough explained how you're going to write and what you're going to write. Okay, good luck for your thesis. Thank you.